Hey there, we're going to just talk about section 17.3 here, which is the pre-mRNA and the splicing of it. So let's first talk about the ends. Let's begin with the ends. We have just finished creating the pre-mRNA strand. It's been transcribed from DNA. And so we need to modify it. And so this is going to be done. First, we're going to put on the 5' cap. The 5' cap is going to go on as soon as the first 20 to 40 nucleotides have been transcribed. So the RNA polymerase is going to start transcribing, and then once it gets about 20 to 4 nucleotides in, the 5' cap is going to be added on. And the 5' cap right over here, as you can see, it's a modified guanine, and it is connected with a triphosphate, or 3 phosphates, to the rest of the nucleotide. So the function of the 5' cap is to protect the mRNA strand as it's traveling from the nucleus to the ribosome in the cytoplasm. There are enzymes in the nucleus, like endonucleases, which take apart nucleic acids. They degrade nucleic acids. So we need to protect our mRNA strand from these enzymes. Next, let's talk about the other end, the poly-A tail. If you remember, on the 3' prime, there was a polyadenylation signal. The polyadenylation signal signaled to the RNA polymerase II with the AAU-AAA nucleotide sequence that in about 10 to 35 nucleotides, that would be the place to cleave the mRNA strand, or the pre-mRNA strand. And so now we have a pre-mRNA strand, and it's being modified. And what is going to happen? So here is our polyadenylation signal. That at the end of the 10 to 35 nucleotides, so here we are, an enzyme is going to add on the poly-A tail, which is just sequences of 50 to 250 adenines to the end of this strand. The functions of the poly-A tail is, well, there's 3 of them. One is that it appears to facilitate the mRNA strand leaving the nucleus. Another is that, once again, it protects the mRNA strand, or the messenger RNA strand, from enzymes that are going to degrade nucleic acids. And finally, it helps the ribosome attach to the 5' end of the mRNA. So when the messenger RNA is attaching to the ribosome, the 5' end is going to attach first. And so this poly-A tail is going to help it to facilitate that happening. So the 5' cap and poly-A tail are not going to be translated into the final primary transcript, which is the strand of mRNA that the ribosome is going to be reading. And so here we have our 5' cap and our poly-A tail. And so this, from our original strand of pre-mRNA, these are not going to be translated into our primary transcript. Neither will the nucleotides around the polyadenylation signal or the promoter region here, so the 5'. We call these the untranslated regions, the UTR. And so what is going to be transcribed starts at the start codon and stops at the stop codon. And so this will be our transcribed section. So we call these UTRs, the 3' UTR and the 5' UTR. All right, so we talked about the ends. Let's talk about the middle, the introns and the exons. So RNA splicing is done by an enzyme called the spliceosome, and the average length of a transcription unit If you remember, that's the entire section that is transcribed into the pre-mRNA. That tends to be about 27,000 nucleotide pairs. So on average, your average protein will have about 27,000 nucleotide pairs in that initial transcription unit on the DNA. However, our final messenger RNA, our final product in the sequence on the primary transcript, is going to have about 1,200 nucleotides, which is about 400 amino acids, because 3 nucleotides are going to code for 1 amino acid. And so that means that there's quite a bit of segments. There's quite a few segments, quite a few large segments of the transcription unit being removed, and we call those introns. We call them introns, and this is confusing because you'd think that we want to keep the introns in and the exons out, but the introns are called intervening sequences. So that's short for intervening sequences, and they're going to be spliced out. And they are spliced out by a complex of proteins, the spliceosome. So that's a number of proteins called the spliceosome. And so the section, so here's our intron, so this is going to be removed. And here's our intron, this is going to be removed. And the sections that stay in are the exons. So here's our exon, and these are going to be spliced together. And then we have our poly A tail and our 5' cap. And so this is going to be the message or the final primary transcript that gets sent down to the ribosome. In this next section here, we're going to talk about ribozymes. Ribozymes are a discovery that was made when scientists found that RNA molecules not only act as messengers, but they could also act as enzymes. So prior to this, scientists thought that the only enzymes in a cell were proteins, and so this was quite a discovery. And so a spliceosome, the complex that we were talking about that takes out the introns and splices together the exons, is a combination of proteins and catalyzing RNAs. And so this was discovered when scientists discovered ribozymes, which are the RNA molecules, catalytic RNA molecules. And a pretty cool fact is that in some organisms, the introns themselves are a ribozyme, and they're going to splice themselves out, and they're going to catalyze their own excision, which is pretty cool. So it's those, remember that the mRNA is RNA, and so it's acting not only as messenger RNA, but it's also acting as a catalyst to remove its own segments of its own self, which, life is so cool. And so there are 3 properties of RNA that allow it to function as an enzyme. And so first, because RNA is single-stranded, it has those free base pairs that, half of the molecule is exposed. It doesn't have that second half like DNA. So those open base pairs are able to base pair with other parts of itself. So if you look here at this molecule, this is a strand of RNA that has folded onto itself. And it can, sort of like protein folding, when you talk about the hydrogen bonds and the intermolecular forces, the hydrogen bonds are going to interact in segments and allow it to fold into itself and create a 3D shape. A second reason that RNA can function as an enzyme is that it has functional groups, or it can have functional groups, similar to how proteins have amino acids, and how each amino acid has kind of its own personality. And so since these base pairs can have functional groups, it allows those intermolecular forces to to react and also to interact in catalyzing functions, catalyzing reactions. Finally, the third reason that ribozymes or that RNAs can act as enzymes is its ability, once again, to base pair. It can base pair not only to itself, but to other nucleic acids. So it could find a very specific spot on DNA, and it's going to add specificity to its ability to catalyze a reaction. It can find a very exact spot because of those base pairs, because they're going to have a very specific sequence that can line up with another very specific sequence on DNA. All right, let's talk about the importance of introns. So we get rid of them, but are they garbage? They're not, and they're still not well understood. There's a lot that we have to learn about introns, but we know that that they do serve functions, and one function is pretty cool. It's called alternative RNA splicing. So if we have our pre-mRNA strand here, we have our red, our yellow, our green, and our blue. When the primary transcript is created here, so we have spliced out introns and we've created our primary transcript, we've removed the green section. And when the gene is expressed, and now the gene can be expressed in another way, so it's going to be transcribed. This is our pre-mRNA, but it's going to be modified a little bit differently. Instead of making the yellow an intron, instead of making the green an intron, it's made the yellow an intron and removed the yellow. And so while it's the same gene, it has been expressed differently. And this is thought to be the reason why humans can have about the same number of chromosomes as a simple organism like a nematode. It's that we can be a little bit more creative. There's more flexibility in how we express our genes. So those introns do serve a function. A second important function is that they put space between domains of proteins. And so let's understand what a domain is first. A domain is sort of like a part of a protein. It is, let's say, the active site or a binding site that will bind to a plasma membrane. And so we can just think of it as a section of a protein. And so let's say that exon 1 is the section of the gene that's going to code for the active site. And exon 2 is the section of the gene that's going to code for the place where it's going to bind to a plasma membrane. And if there were no introns, those two exons would be together, close together. And during recombination or crossing over, it would be a higher likelihood that that exon is going to be interrupted. Those important segments that create a portion of the molecule of the protein are going to be split apart and rendered useless. And so these introns allow for sections right here for those entire sections of the exon to be able to cross over, so that you'll get the full active site or the full binding site for the plasma membrane. And so this allows for shuffling around, and which also allows for variation. So if you're going to add a new active site or add an active site to another version of this gene, well, this might create something totally brand new. And we call this variation, and this is a key concept in how a species changes over time and what allows us to adapt to changes in the environment. And so that's all we're going to talk about for today. And 17.4, or translation, will be up next.